to tell you about a remarkable story that we've discovered at Metys Pharmaceuticals. My name is Mike Schertz. I'm the founder and CEO of the company. We are a group of people who formerly worked together at Actelion. I'm the sole founder and the sole shareholder of the company. We've received 3 million Swiss francs in seed investment capital, mainly from former colleagues of mine at Actelion, some of them founders of Actelion. That seed investment is all structured as a convertible loan, repayable at a 20% discount to the pre-money valuation that we agree with the Series A investors. We are developing MP101, which is a small molecule, a very small molecule. It acts in the brain and in the spine as a modulator of glutamate signaling. It's orally active in rodent models of depression, of cognition, and in neuropathic pain. In neuropathic pain, we have single dose and chronic data as well, but the depression and the cognition data we have are only single dose data. The compound has a benign safety and tolerability profile in up to 200, a total of 200 patients worth of clinical data of the racemic dimiracetam. The toxicology, the pharmacokinetics are all well behaved. The compound has ideal pharmaceutic properties. And I'll tell you a remarkable story about the fact that this molecule, as its racemate, has been developed twice for cognition the first time and for neuropathic pain the second time. Chemotherapy-induced peripheral neuropathy and diabetic peripheral neuropathy are big, important markets for neuropathic pain drugs. MP101, with a complete and ICH-compliant regulatory dossier, is ready for phase 2 trials in both indications. This is dimiracetam. It's a very small molecule, has a molecular weight of 140. I think it's smaller than 95% of any drug that's ever been approved. It's tiny. And it was developed twice as its racemic mixture, first by SmithKline French and SmithKline Beecham in their laboratories in northern Milan. Because the compound, it's a rigid analog of piracetam, so the my pointer here, this carbon-nitrogen bond is missing in piracetam. And Carlo Farina, the chemist who made the molecule, he was looking for more potent versions of piracetam, so he tied the ears back. He made a rigid analog, which is 300 times more potent. And because the pharmaceutic properties are ideal and the compound is toxicologically benign, SmithKline Beecham developed the compound as its racemate all the way to phase 1. The CMC, the ADME, the tox, clinical trial data, single dose data, and then Glaxo closed the site. They closed down the research facility in Milan. And they took all of the reports from the study and they threw it in the dumpster. Literally. Carlo, the chemist who made the molecule, climbed in the dumpster and took the reports home. We have today the full set of original SmithKline French documents from 1991, 1992, 1993, from the clinical development, the toxicology, the ADME, the CMC, the whole package. We have it all. When the composition of matter patent expired in 2006, Carlo filed a new method of use patent for racemic dimiracetam, for the use of dimiracetam for the treatment of neuropathic pain. And with the money he had as CEO of NICAM Research, a medchem services company, they redeveloped the molecule. They redid, Glaxo wouldn't give them the original data, so they redid all of the CMC, all the ADME, all the tox, all the phase 1 data. They sold it to a Swiss company who ran a phase 2 trial in HIV patients in rural South Africa. The study is worthless, uninterpretable, and they stopped it. And that's the package that we licensed back then. When we created Metys Pharmaceuticals, we were hoping to find new investors who would invest in a method of use patent of racemic dimiracetam for a real clinical trial of racemic dimiracetam. Nobody was interested. Nobody. We tried hard for 2.5 years. Nobody was interested. I gave up. And I sat in my office on a quiet Saturday morning, looking through the old data, and there was a weird experiment. Both times that dimiracetam had been developed, it was developed as its racemic mixture. Why? Why would you do that? Why would you twice develop the racemate? It turns out the S enantiomer, this molecule here, is almost inactive. The R enantiomer is the active enantiomer. But remarkably, when you make the racemic mixture, the one-to-one mixture of those two molecules, it's more potent, a lot more potent than the R. And the R more potent than the S. And that's why, twice, they developed that racemate. And by the way, this phenomena is not because of the pharmacokinetics of the two enantiomers. The two enantiomers have superimposable pK. If you give the racemate and measure plasma concentrations of the two enantiomers independently, they have superimposable PK profiles. There's no funny business. It's not like the S enantiomer is inhibiting the metabolism of the R or something like that. The question we asked, and the question that had been asked when I was looking through those papers on that quiet Saturday morning, where in this curve between pure S and pure R and the racemate, where's the maximum in this curve? Is it in the middle? It doesn't have to be. And so we went looking. We went looking for ratios of 2 to 1, 3 to 1, 4 to 1. When you make a 2 to 1 or a 3 to 1 mixture of these enantiomers, it's 20 times more potent than even the racemate. This is weird beyond description. It's also patentable. And that's what we've done. We've patented these non-racemic mixtures between 2.0 to 3.2 R to S. Here are the data. We had known for quite some time that racemic dimiracetam is effective in a rat spinal synaptosome preparation with an IC50 of about 20 nanomolar. That is, a preparation where you take the spinal cord out of the rat, throw the rat away, you keep the spinal cord, and you homogenize it incompletely, carefully, filter it. And these nerve endings, they're still pharmacologically alive. You can load them with tritiated neurotransmitter, tritiated D-aspartic acid, and they actively transport and store it inside of the synaptosome. So when you now filter and wash away the excess radioactivity, the synaptosomes are laying on the surface of the cell membrane. And when you now add NMDA and glycine, they're let loose. And in the filtrate, you get a spike of radioactivity. If you do this in the presence of racemic dimiracetam, it inhibits that release with an IC50 about 20 nanomolar. So we went to Professor, he'd published this back in 2009. We said, please do the experiments again with racemic dimiracetam. The IC50, 15 nanomolar, good enough. Now the two individual enantiomers, unlabeled. The S enantiomer, 420 nanomolar IC50, the R enantiomer, 123. That fits. That's the same thing we had seen in those earlier experiments. Let's try it again with the non-racemic mixture. The IC50 of the 3 to 1 R to S mixture was less than a nanomole, about 0.8, factor of 20 more potent. In vivo, in a rat model of oxaliplatin-induced peripheral neuropathy, we see the same very unusual medicinal chemistry behavior. The S enantiomer is almost inactive, the R enantiomer is more potent, the racemate is more potent still, but MP101, the 3 to 1 R to S mixture, is even more potent by a factor of 5 at least. We have a lot more data that are all embedded in the patent applications, and the patent application in America is just published. You're welcome to have a look at that. I'm happy to share it. So the thrust, of course, is that this is something that we can patent, but it also teaches something about how the molecule is working. It's modulating glutamate-induced glutamate release, which teaches you that you would then use it in a number of different neuropsychiatric indications or motor neuron disease indications. The question we want to know is whether all of the phase 2 clinical data of the racemic material Can I use that again? Or do I have to redo the tox? If we want to run the clinical trial of this newly patented 3-to-1 R2S mixture, can I use all of that data? Or do I have to redo the toxicology? Well, the FDA, in their guidance for the development of seroselective development of drugs, have said, quote, a relatively benign toxicologic profile using the racemate would ordinarily support further development without separate toxicological evaluation of the individual enantiomers. Almost. When I've shown this to big pharma and to big VCs, they've all said the same thing. That's nice, that's encouraging, but please bring it to us in writing from the FDA. The guidelines are one thing, but what they're actually going to do may be something different. So we've applied for a pre-IND meeting, and I am thrilled to say that they have accepted us for a face-to-face meeting. We're going to go see them on the 27th of February to ask this question. There's some other questions we're asking as well, but this, I think, is important. Whether they will accept it. If they do accept it, without any bridging studies, we have all of the phase 2 data to support phase 2 clinical trial of MP101 in an indication of our choosing. How can this be? How can you explain this? And I'll show you an explanation that I think is real. What I'm going to show you is incorrect, but it provides at least a plausible explanation of what's happening. In 2010, an academic group, we have nothing to do with Bob Oswald and his group at Ithaca at the time that we discovered this paper, they published the X-ray crystallographic structure of the orange section. This piece here, which is taken from one of the glutamate-gated ion channels. Glutamate activates a whole family of different ion channels. All of them have a glutamate recognition domain, this orange section, which is common to all of them. This one happens to be from the AMPA type glutamate-gated ion channel. They crystallized this orange section, and they soaked those crystals in a solution of piracetam, this molecule here. Remember, dimiracetam is just the rigid analog. They've published the chemical structure. It looks like this. Ooh, look, it's not one molecule of piracetam. It's 6. 6 separate molecules of piracetam in that X-ray. It's not fair to say 6 because, in fact, the whole thing is rotationally symmetric. It has a C2 symmetry axis. There are 3 unique sites just on the left, or the ones just on the right. It's the same thing. It's rotationally symmetric. So we've taken those 3 unique sites into a computer model, and we've tried to fit dimiracetam onto the X-ray crystallographic data from that paper back from 2010. When you do that, the R dimiracetam fits beautifully for 2 of the 3 sites, not on the 3rd site. For the 3rd site, only the S enantiomer fits, but not on the other 2. That's a 2-to-1 ratio. It's wrong. I know it's wrong because it's the AMPA type receptor. And we're interested in NMDA. AMPA is a homodimer, NMDA is a heterotetramer, But you get the idea. It's like oxygen binding to hemoglobin. Remember? You learned it in graduate school. The first one is slow, the second one is a little bit faster, the third one is faster still, and then zip, it's a cooperative binding event. Something similar, I think, might explain this behavior with dimiracetam. In other crystal structures from NMDA and AMPA type receptors, there are positive and negative allosteric modulators of these glutamate signals binding to exactly the same analogous location in those crystal structures. But those molecules are big. They have molecular weights of 360, 420, 280. You can't fit those molecules in like you can with this very small molecular weight of 140. We have material transfer agreements with Professor Oswald, he nearly dropped the phone when I first told him the story, and Professor Stephen Trainellis at Emory University. Both of them, we've sent the samples. They're doing X-ray crystallography and electrophysiology. I've not gotten involved in what they're going to do. We're going to see whether it's true or not. But it should give us an idea of how this phenomena can come about. We also have filed patent applications for related compounds that show the same behavior. It's not just dimiracetam that does this. Certain structural analogs also have the same behavior. Let me, in 2 minutes, tell you about the clinical trial program. In neuropathic pain, dimiracetam is effective in treatment and in prevention. This is the prevention study in rats who are receiving, in blue, intraperitoneal infusions of oxaliplatin. Racemic dimiracetam at a low dose of 50 milligrams per kilogram twice a day, or at a very low dose of 15 milligrams per kilogram twice a day, can prevent, to a certain extent, these symptoms. If we gave higher doses, we could prevent it completely. We give the same dose on a milligram per kilogram basis of MP101, this 3-to-1 R to S mixture, and the protection is complete. This is what our clinical trial data should look like. Chemotherapy-induced peripheral neuropathy is a big unmet medical need. Won't go into it. What people often forget about chemotherapy-induced peripheral neuropathy, it does not start as pain. It's not, at the beginning, it's not painful. It's a cold sensitivity. In this study published by Professor LaPrince, he's asked patients to score on a scale of 0 to 10 their sensitivity to touching cold items. This is the primary endpoint that we have chosen for our clinical trial of MP101, cold sensitivity. After a period of 6, 7, 8 infusions, this turns into a pain syndrome, which will be our secondary endpoint. But in the phase 2 clinical trial that we're going to conduct with the funding that we're looking for, we will measure as a primary endpoint cold sensitivity around the 4th or 5th cycle of a 6 or 12 cycle FOLFOX regimen. If it hits the pre-specified statistical hypothesis, we'll then test the middle and the low dose. We'll also run a phase 2 clinical trial, a proof of concept study in diabetic peripheral neuropathy, and that's a randomized trial withdrawal design. The tranching is illustrated here. We'll start with $4 million with the safe to proceed decision come the major investment, 17 for the conduct of the main trial and parallel studies, and a third tranche for the diabetic peripheral neuropathy study. We're happy to talk with anybody who can help us find the money to do this work. Thank you for your time, and look forward to any questions you might have.